Hi everyone, today is April 15th, 2020. My name is Mara Burrows and we are doing Bio 225, week 6. This is lab 9 and we are doing a salt tolerance and pH testing. This video specifically is for the salt tolerance, so I already have all my stuff with me. I have my salt broths. There are 6 in total. They are all labeled either S-Epi or S-Seri, one of each. I have my broths. I also have my tea candle and my pipette that's in alcohol. So let's start the inoculation. So I'm going to take my pipette, I'm going to dry it. First, we're going to start with the S-Epis. So I'm going to take my 1% S-Epi, which is this one right over here. I'm gonna open it, put it over the fire. Next, while that's open, I'm gonna take my S-Epi broth, put this over the fire as well. I am gonna use my pipette to get enough for 2 drops. One, 2. Before I close the salt broth, I'm gonna go ahead and put it over the flame again. And close it. I am going to go ahead and do that for all of the S-Epi and then do it again for the S-Seri. Once that's done, we're going to inoculate the broth for about 48 hours. If it is a positive, it's going to turn out turbid. If it's negative, it'll still stay clear. So we'll take a look at how it looks like in 48 hours. Okay, so we finished the salt test. Now we're going to start the pH test. I have 6 vials of the nutrient broth already. I've already labeled them pH 5, pH 7, and pH 9. 3 vials for S-Epi and then 3 vials for the S-Seri. I have my nutrient broth with me, my candle, my pipettes that are in alcohol. I also have the sodium hydroxide and the HCl, hydrochloric acid. So to start, we're going to take the S-Epi pH 5 tube, which is this one right here, S-Epi pH 5. I'm going to go ahead and open it up and I'm going to pass the opening over the flame to sterilize it. Just like that. Next, I'm going to add exactly 1 drop of the hydrochloric acid into the tube. Just do one drop. Okay, 1 drop. Next, I'm gonna put it over the candle again, and we are now gonna close it. So after that, I am going to get the S-Seri pH 5, which is this right here. And again, I'm going to do the same. Open, go over the flame. 1 drop of the hydrochloric acid. Okay. And I'm gonna, oops, before I do that, my mistake, go over the tube to sterilize. And then close. All right. I'm gonna set this aside for now. The pH 7 broths, we are not going to do anything with. Those are already adjusted to pH 7, so we don't need to add an acid or base to that. Next, we're going to do the pH 9s. The pH 9s, we're going to also add 1 drop of the sodium hydrochloride, or sodium hydroxide, sorry, into each vial. So we'll start with the S-Epi, pH 9. I'm going to cut this open. Over the flame. 1 drop. Again, this over the flame. Close. Okay. I am going to do that with the S-Seri, pH 9. Same thing. One drop. And again, over the flame. Close. Okay. I'm going to go ahead and wrap these pipettes in a paper towel, and I'm going to place it in the trash. I have this here. We're just going to toss as is. I'll put this aside. Okay. Now I'm going to take a graduated pipette out of the alcohol cup, and I'm going to go ahead and shake it up to make sure it dries. Next, I'm going to take the S-Epi broth, and I am going to use aseptic technique. So I'm going to take all my S-Epi that I put in, and I'm going to put 2 drops in each. Let's start with taking this over the fire. Sorry, forgot to put it over the, close it. Okay. Next I'm going to take this, again, oops, that out. I'm going to put 2 drops, 1, 2. Shake it up a little bit. And I will do the rest for the rest of the S-Epi. I'll also do the rest for the S-Seri, and we will go ahead and incubate it for about 24 hours, and then we will see the results. Thank you.